I'm Dr. Xu from Sun Yat-sen University Cancer Center, and it's my great honor to represent my group to share our discussions here. But first of all, please allow me to speak in Mandarin. That will make me easier and save time. Please. Thank you.
My group discussion based on Tivatinib used for GI cancers in the second phase study. Our discussion is based on the available information and data we obtained. We feel that there is a lack of sufficient data that is available to us. Our first step is that we need to select the right kind of tumor. So, based on our data, in the preliminary stage of the clinical trial, we can have some cell line with strong implication of c-MET for lung cancer and gastric cancer. So after discussion, we decided to choose gastric cancer based on several reasons. First, in the preliminary clinical research, c-MET can see its positive representation in the gastric cancer cell. Secondly, in China, we can find a large population of gastric cancer patients. Number three, currently, we are not sure about how effective c-MET is for gastric cancer. But for gastric cancer patients, it's easy for us to get tissue sample for biomarker analysis by endoscopic biopsy. So that's the reason why I choose gastric cancer as the target of our discussion. But we're also faced with some challenges. So based on available information, the medicine is relevant to c-MET. But the c-MET positive rate was not clear in Chinese gastric cancer population. And the medicine relevance with the mutation is not clear to us also. Another major issue is that the phase 1 and early phase 2 data is not sufficient concerning gastric cancer patients. Because this medicine is oral taken dose, because gastric cancer surgery and the tumors factors may affect the absorption of the medicine. So for the PK information about this group of patients is not sufficient either. These are the problems that we need to consider in the second phase research. The step 2, the selection of study population. So we decided to choose patients who have advanced and metastatic gastric cancer. We also had heated discussion as to whether we should choose all gastric cancer patients regardless of the metastatic condition, or whether we should focus on the metastatic gastric cancer patients. After discussion, which is quite heated, we believe that although the preliminary clinic process information may indicate that the efficacy of the drug may be relevant to c-MET, however, we do not have enough information concerning gastric cancer, and as for the abnormal of c-MET may express in what kind of form, whether in overexpression or mutation, is not clear to us. So finally, we decided to use the design of early phase 2 design. We decided to make the exploration and we decided to choose all patients despite of CMET situation. And for the selection of study population, because they are all advanced gastric cancer, we also discussed as to should we develop the medicine for the first line, second line, or third line. Considering that there are established first line and the second line basis in China, and we have already reached quite a lot common understanding. So based on the ethical consideration, we decided to make the choice of third line medication. But because of its third line, another issue was brought up. Group members mentioned that after 2 times of chemotherapy, the general situation, physical condition of the patients may be quite poor, and the overall survival period may be relatively short. Therefore, we also discussed on the endpoint selection. Some group members mentioned that we need to choose overall survival as the endpoint, because for the third line of the patients, the survival period is quite short. So if we chose overall survival as the endpoint, we may have better opportunity to observe the efficacy of the drug. Even if the objective efficacy may not be represented, we can observe the benefits brought by the agent to patient. However, if we want to measure overall survival, we need to have another control group, which inevitably means that we need to enlarge the population of the patients. And also for the early study, we need to focus on whether the agent is really effective for gastric cancer patients, because information is too limited, and it's highly possible that this medicine is not effective on the patients. So finally, we decided to choose RR as the endpoint. Step 4, we discussed on the study design. Should we choose randomized control study or the single-arm study with Tivatinib, while all patients would receive the positive agents for treatment? Because we choose overall survival rate as our endpoint, we finally chose the single-arm study. Because we did not know the status of cement, we hope that all patients can receive treatment, and we can have more in-depth follow-up treatment to analyze the total efficacy of and the effectiveness of the agent. Jack Lee then helped us to design the sample size. The assumptions include the preliminary estimation is that CMET positive rate in all gastric cancer would be 30%. And among the CMET positive patients, based on clinic discussion, we could have the efficacy rate of 52%. Then the assumption is that the effective rate among CMET positive patients would be 50%. And the p-value is 0.05, and we need to have 60 patients in our sample. So then, I would like to brief you on our inclusion criteria. We've already defined it as the third line. We need to have measurable criteria for the patients. So we need to also have measurable lesion after the second line of measurement. The patients only to have the second line assessment. Because this is third line treatment, we worry about the overall survival time. So we require that the patients need to have a relatively good ECOG performance status. between 0 and 1, and the expected survival could be 1 to 3 months. Exclusion criteria. We emphasize that if these gastric cancer patients have received surgical operation, or if the oral taken of the drug would be affected, we would exclude those patients. Safety is another consideration for the phase 1 study. This is not quite popular among gastric cancer, so we need to focus on safety a lot and exclude all other possible negative factors. As for the dose selection, we do not have any detailed information yet, so we'll depend on phase 1 studies recommendation. And additional issues for the design of the second phase research, we need to collect more individualized PK data concerning the gastric cancer patients in China. And also in our design, we need to optimize the selection and the validation of the biomarkers. Therefore, for all patients, we need to have PK tests, especially for the first 10 to 20 patients. The PK tests need to be done in a more careful way. After getting the thorough information of PK among these patients, if the information is quite similar with other types of cancer patients, only based on that can we carry out the next step research. And also, for all the patients before treatment, we need to have the sample of the we need to have the tissue by biopsy through endoscopic treatment. And the analysis of biomarker also includes many aspects, including the expression of c-MET and the mutation, c-MET mutation, and the clinical function test also need to be done in a more extensive way.
Thanks for all members from our group.
So if I miss anything, I welcome my group member to supplement my presentation. So we have time to ask just one question.
One question?
I'll ask a question. Do you think a 50% response rate is realistic for a third-line agent? I don't know how to say this in a...
Maybe it's a little bit high, but the only data we get from our materials, that's 52 response rate in the data.
Answer that question. Actually, the Simon's two-stage design was based. You need 4 parameters, and I know because yesterday, even though I did the calculation, but the time is short, you probably didn't capture everything. So we need 4 things. We need alpha, beta, P0, and P1. Okay. So alpha is 0.1, beta is 0.1, P0 is 5%, and then P1 is 15%. How do we get 15%? Is because 30% of the patient will have c-MET positive, and in that group, we assume the response rate is 50% in that subgroup. So overall, it's 15%.
15%.
So we did not design the overall group, with a response rate of 50%. The overall group, the response rate is 15%. Yeah. So that's sort of, the clarification.
Thanks.